Hello everyone, my name is Vaijayanti Gupta and I'm a senior scientist here for Strand Scientific Intelligence. Today I would like to talk to you about our new product, Avadis NGS, which is our solution for the analysis of NGS data. Avadis NGS is an application for the analysis of next-gen sequence data for RNA-Seq, DNA variant analysis, ChIP-seq, which are all provided as separate modules, and all of which are relevant to the understanding of cancer biology. This is specifically designed for and by life science researchers. In the next few minutes, I would like to show you how the product works and is useful particularly in looking at sequence data from various cancers. How it can help you answer real biological questions without having to be an expert bioinformatician. Avadis NGS provides downstream or tertiary analysis for NGS data, taking in pre-aligned data. Primary analysis is the analysis which is performed by the NGS hardware platform that controls the machinery that does the actual sequencing. This generates the raw reads with associated quality metrics. Secondary analysis takes the reads from the primary analysis and aligns these reads to a reference genome. After the primary and the secondary analysis steps have been performed, the data is ready to be loaded into a tertiary analysis program, and Avadis NGS is one such. Avadis NGS will take in the pre-aligned data, usually in the SAM or BAM format, and it will allow you to interrogate the data in a structured and interactive manner. There are three major modules in Avadis NGS. These are RNA-Seq for the transcriptomic data, DNA-Seq for variance analysis, and ChIP-seq for transcription factor and other protein binding sites. Although all three modules are relevant to cancer biology, we will primarily focus this presentation on the RNA-Seq and the DNA-Seq modules. Avadis NGS supports the classical differential gene expression analysis using normalized expression values in the RNA-Seq module. At the same time, the user can use the same data to interrogate alternatively spliced transcripts, SNP calling in the genes, which are all relevant to biology of cancer. In many cancers, the genetic makeup of a cell is changed in such a profound way that two genes can get fused together, resulting in a hybrid protein with potentially devastating effects on the cell. These gene products can be further detected with Avadis NGS, and we will show you examples in this presentation. The DNA variance analysis workflow, which is geared towards the detection of SNPs, small insertions and deletions or InDels, and large structural variations will also be demonstrated. SNPs and InDels are discovered and annotated with information from the database, the dbSNP NCBI or equivalent databases. The workflow also contains features for large structural variants. These structural variations like large insertions, deletions, translocations, and other rearrangements can be detected in these experiments using paired end or mate paired libraries. As I mentioned today, we will first talk about the RNA-Seq and the DNA-Seq module in today's webinar. What are the main questions that a cancer biologist would like to see in the data that's being generated from next-gen sequencing? A classical study would be to look at differential gene expression between cancer and normal or paired normal tissue. In addition, many genes undergo alternate splicing. Some of the questions that are being asked in the cancer world today are whether some of these genes have alternate transcripts indicative of the tissue of origin of the cancer, what cancer-causative mutations are inherited and which ones are acquired, how do these mutations affect genes or the encoded proteins. The hallmark of cancer is often large chromosomal rearrangements, deletions, and copy number changes. Studying these are important in the understanding of cancer. Can we detect such large changes using the data? Another interesting phenomenon that arises from various rearrangements that happen in the cancer cell are various fusion proteins or read-through transcripts. Can we detect these using the data? And finally, what are the signaling pathways and networks that are affected in cancer? To start with, the data needs to be imported into Avadis NGS. We have chosen to support the SAM and BAM format as the main import format for pre-aligned data, and this is the de facto standard. Avadis NGS pre-packages annotations from humans and other model organisms. These pre-packaged annotations include reference sequences, dbSNP IDs, transcript annotations, and so on. In addition, various versions of genome annotations are updated at regular intervals and made available to the user. You can update these by using the annotation update feature of the tool with a couple of simple clicks. The important point here is that this is one feature that Avadis NGS is very useful to the user in the fact that these annotations will be available to the user at whenever they're ready. The import wizard launches upon the creation of a new experiment. You can specify the organism of choice, human in this case, and the version of genome annotation that was used to align the data. You can choose the transcript annotation database from amongst 3 available transcript annotations: Ensemble, REFSeq, and UCSC. Align sequence data from a variety of platforms are supported. Illumina, ABI Solid, Roche, all of these are supported by Avadis NGS. The type of sequence data is specified under platform, as you can see here. In addition, the user also needs to specify the type of library that was used to generate the sequence data, whether it was single fragment or paired end or mate paired. The latter are especially important in the detection of large structural variations. It is important to note that if your sample is split over multiple files can be given the same name and you can aggregate all the reads into a single sample. Here we are importing data from the analysis of melanoma transcriptome by Berger et al., which appeared in Genome Research in 2010. In this experiment, we have created there are 3 files for melanoma cell line and 2 for leukemia, and we are aggregating them into a melanoma sample and a leukemia sample, as shown here. Once the data is loaded, there are some common steps that one has to go through to analyze the data, particularly the quality. The figure shown here in the slide shows you the QC plot and the heat map that tells you about the sequence quality of each tile. The QC plot denotes tiles on x-axis and counts on the y-axis. The higher count for the pink spot means that most of the reads in the tile map will unambiguously map to the reference genome, and they are all of high quality. If the count for pink spots drop and consequently the number of red or blue spots are higher, it implies that for those tiles, most of the reads will map to multiple locations on the genome. To interpret the heat map, again, the tiles are along the rows. The red on the left-hand side denotes that the reads are of very high quality in the beginning, and gradually the quality falls by the end of the read, as denoted by yellow and then gray. Occasionally, you will notice tiles where the color is gray or yellow patch in the beginning itself. This indicates that the sequence from the tile are of low quality at the beginning of the read, and you may want to discard some of these from your analysis. The user can now use filters to filter out these bad reads, and these filter options are available in the workflow. Once the data is imported and QC is performed, we can now proceed with the downstream analysis depending on whether it's transcriptomic, exome, or whole genome data from various cancer tissues. We will first describe the RNA-Seq for the transcriptome analysis. What questions in cancer biology can the transcriptomic data address? Well, one can look at differentially spliced genes, which is the most common use. One can look for alternatively spliced transcripts, novel fusion genes, read-through transcripts, somatic and germline mutations in genes of interest. All of this can be addressed using the RNA-Seq data. In the step 1, what is done is read counts are converted into normalized reads per million sequence per kb of the gene, or RPKM. And then these RPKM are compared between 2 classes of samples. Simultaneously, there are a large number of visualization aids also available in Avadis NGS to visualize the results of the differential expression. Here you can see a volcano plot that plots the relative up and down fold changes in expression and plot them to the negative log of p-value obtained from the significance test, such as t-test or ANOVA. The spreadsheet here will list the absolute fold change and corrected p-values from various statistical tests. The user can set filters and create gene lists depending upon what fold change value and p-value cutoff one decides to use for the spreadsheet. In addition to the volcano plot, another useful view that's present in Avadis NGS is the profile plot. This displays the relative expression level of genes in each sample within a group. In this example, the genes that are relatively upregulated in leukemia are shown here related to the melanoma samples which are to the right. You can see that the genes which show a higher expression in leukemia, the leukemia samples are to your left, and these genes show relatively lower expression in the melanoma samples, and this is to your right as indicated by the profile plot. Once you're done selecting a list of genes, then the next step is to perform an analysis that will tell you about the biology of these genes. So once a list of interesting target genes is constructed, the annotation of those genes can be evaluated using GO or gene ontology function and the pathway analysis function in the same tool. However, we all know that it's not a gene that's expressed, but its constituent transcripts. Alternate splicing events are found in a high proportion of cancer cells, either as a consequence of cellular abnormalities in cancer cells, or more importantly, as the cause of cancer growth. In Avadis NGS, a special view has been developed to study differentially spliced genes. The result of a typical analysis is shown here. We have developed a special gene-centric visualization to display the differential splicing results and corroborating evidence for this claim. And this view is called the gene view. The gene view will show you all the transcripts, both known and novel, for a particular gene. Here is an example for the gene GLRX. There are two transcripts for this gene according to the RNA-Seq transcript model, which differ only in the length of the first exon, as you can see here on the left-hand side. The profile plot shows you three different lines. One in red, which shows you the RPKM value for the gene, the entire gene. The blue and the brown show the RPKM values for the two individual transcripts. The expression level of the blue transcript stays basically the same in both leukemia and melanoma samples. The brown transcript goes up significantly in melanoma samples compared to leukemia samples, as you can see here, leukemia to the left and melanoma to the right. The evidence, so as a consequence of this, the relative expression of the gene is also high in melanoma compared to leukemia. The challenge in this analysis is that for a whole gene, all exons within the gene structure will be considered. The transcript read counts need to be recalculated for each individual transcript, depending upon the constituent exons that are included in the transcript structure. Here's an example from the leukemia melanoma data that we talked about. An example of this is a gene, RAP27A, which is a small rat-like GTPase superfamily. It is identified as differentially and alternatively spliced gene between the MEWO melanoma cell line and the K562, which is the leukemia cell line. There are 4 transcripts for this gene, as you can see above in the little blue bars that indicate the transcripts. As seen from the profile of the extended exon on the right-hand side, which is present only in transcript B, it is downregulated in leukemia compared to the melanoma samples. Consequently, transcript B will be downregulated in leukemia compared to melanoma samples. Conversely, the last exon on the right-hand side is present only in transcript D, which is upregulated in leukemia compared to melanoma. Transcript C should be downregulated in both leukemia and melanoma, as evident from the expression values of the last exon on transcript C. Thus, we can see how amongst 4 transcripts for a gene, we can identify one that is up one that is down, and one that has no change in expression for the same set of samples. In this example, it is easy because there are unique exons to each of the transcripts. But if you don't have unique exons, but only unique splice events, one can use additional hints. For example, you can look for unique reads that will be spliced across exons, which will be unique for a particular transcript. So we go back to this example of the small GTPase encoding RAS gene, RAP27A, with 4 different transcripts. Although the 4 transcripts are reported in this gene, both in On3 and UniProt, there is no report of alternate transcripts being associated with one or more cancer. In this data from the RNAseq experiment, what we are finding is that one of its transcripts is up in leukemia and one in melanoma, and 2 of them don't show any relevant change. We show the profile expression of each transcript that corresponds to a particular condition. The overall gene count is only marginally higher for leukemia compared to melanoma samples, because the overall gene count has the contribution from all 4 transcripts. However, when you inspect at the individual transcript level, transcript D is, as predicted, is higher in leukemia compared to melanoma, and the converse is true for transcript B, while transcript A and C have low level expression in both samples. And you can interrogate this exon by exon. As you can see, the relative expression of one particular exon is higher in one sample versus the other. The leukemia cell line in this experiment K562 harbors the classical gene fusion BCR-ABL, which produces the chimeric protein, the BCR-ABL1, which arises out of the chromosome 9, chromosome 22 translocation, and it is known as a hallmark for leukemia cells. Avadis NGS can detect many such fusion proteins from paired reads in the leukemia sample. Several read pairs for the BCR gene on the chromosome 22 map to the location of the ABL gene in chromosome 9 and vice versa. So if you have sufficient number of paired reads which are in 2 different genes, then you would think that it is a gene fusion. The user can actually right-click on the track showing the reads for each sample, set the filters, and only see those that are translocated reads. You can also detect gene fusions if the 2 ends of the paired reads map to 2 different exons assigned to different but adjacent genes, or what we call as read-through transcripts. In cancer cells, where there is massive aneuploidy and chromosomal changes, often an intergenic region can be deleted in a chromosome. This may result in exons from different genes coming in close proximity and resulting in a new read-through transcript, hence a fusion protein, which is otherwise absent in a normal cell. The read-through transcripts are denoted by the solid lines in the Genome Browser in Avadis NGS. Paired reads are designated by dotted lines. So you can detect these dotted and solid lines in the chromosome and identify these events. If you have sufficient evidence that a paired read spans 2 exons and are spliced, then these would indicate read-through fusion genes. A well-known example in cancer biology is that of TMP-RSS2 ERG gene fusion, which is caused by interstitial deletion of chromosome 21 and is a prognostic marker for prostate cancer cells. A more recent example of a fusion protein is ELM4-ALK fusion that arises from a similar gene inversion on the short arm of chromosome 2 and results in a chimeric protein which is found in 4 to 6% non-small cell lung cancers. What is even more interesting is that this fusion protein responds to tyrosine kinase inhibitors and leaves patients with a great chance for targeted therapeutic cure. The story of EML4-ALK fusion is even more interesting in the fact that recently there has been some patients who show resistance to targeted therapy. In fact, it was using Illumina deep sequencing cDNA results from patients' tumor that revealed that there could be 2 additional and independent mutations which result in structural changes in the protein that is transcribed, and it interferes with EML4-ALK's ability to bind the targeted drug. Indeed, mutations in the exons that can potentially damage a protein are important to the cancer biologist in many respects. In Avadis NGS, the RNA-Seq workflow will include SNP analysis features for the RNA and predict consequences of these polymorphisms on the proteins. Here we are looking at a zoomed-in region of an area where the reads are shown as gray bars with only the mismatches to the reference genome showing up as base calls. We will talk about the SNP analysis in more detail in the next section, which focuses on DNA variant analysis in cancer biology. But the major functionalities of the SNP analysis features remain the same both in the DNAseq and the RNA-Seq workflows. Of course, for cancer biologists working in NGS data, a key set of functionalities would lie in the analysis of whole genome or exome data from cancer tissue, compare it to blood or normal from the same individual, or normal members of the same family, or even another normal person. These various comparisons would help identify structural variants and small polymorphisms in the genome which are critical for tumor development, or those that are inherited, and so on. We will talk about various available functionalities in the DNAseq module of Avadis NGS that will aid the cancer biologist. In the case that the experiment contains multiple samples, a special SNP report is created that represents each of the position for which at least one of the samples shows a SNP or an InDel, and the call that was made for each of the samples. Let us consider an experiment where we compare blood and tumor tissues, paired samples from cancer patients, etc. The SNP detection step will generate a multi-sample report. This combined spreadsheet lists all the SNPs that are detected in at least one of the 6 samples. It denotes which sample and the polymorphism and various other information for that SNP. In this example, the SNP was detected in sample 1, C to T heterozygous SNP. But the second sample did not show any evidence for a SNP here and was called as reference sample or REF. In the second example, a homozygous TT SNP was found in both samples, though the score for both of them is fairly low. SNPs and the DB SNP IDs are called out as known SNPs. After the filtering step, the SNP detection workflow step can be run, and a typical example of the result is shown here. The table shows a lot of information for each SNP that was detected. The first row shows SNPs for which there are 21 supporting reads from a total of 21 reads. This makes it a homozygous SNP. The next column shows that the SNP had a score of 89. A higher number for this score would provide more confidence that this SNP is real. The calculation of the score is a complex algorithm but takes into account things such as number of reads supporting the SNP versus the reference, the quality score for the base call at this position in each read, and the mapping quality of the read and a number of such other criteria. The SNP is also a known SNP since it's annotated with the DB SNP database identifier. The next highlighted row shows an area where there are more than one consecutive position which is different from the reference, and this is known as MNP or multiple nucleotide polymorphism. The MLP here seems to be a novel discovery, as concluded here in this particular column. Small insertions and deletions can be detected using Avadis NGS, and this will also be indicated in the spreadsheet. In addition to the information for the SNP itself, the table also contains information on the possible effect of the SNP or the InDel, which it may have on coding regions if it happens to fall into one. Here is an example that a SNP that would result in a non-synonymous change in amino acid. You can read that in the consequence column, which changes a lysine to an arginine residue for a particular transcript. Here we show you an example of a SNP detected in both blood and tumor tissues. You can visualize it using the Genome Browser, which happens to be the central pane of the tool. The SNP has a G to A transition. You can verify that it is indeed a SNP from inspecting the read tracks for both samples, where the reference base G is altered to A. You will also notice that the SNP is represented in a column of only green. This indicates it's a homozygous SNP. And indeed, the SNP detection region lists tracks for both the blood and the tumor samples. It indicates that it's a homozygous SNP. Mouse over the SNP in either track to find details such as whether or not it's a known DB SNP or a novel SNP, how many reads support the SNP, what is the decibel score, etc. Since it is present in both blood and tumor samples, it's probably a hereditary change. What is also informative is that the SNP falls in the 5' UTR of the gene NTRK1 as shown in the UCSC transcript tract. The SNP detection workflow, as shown in the previous slide, allows for the detection of small insertions and deletions, typically of the order of few bases. But large insertions and deletions can be detected with Avadis NGS while using the paired end or mate pair data as demonstrated here. The colored blocks represent different locations on the chromosome or genome of a single sample compared to the reference sample for the species. The sample on the left represents large insertions compared to the reference genome, while the sample on the right represents a larger deletion. If a paired end or mate paired library is created for these samples, the mate pair fragments will roughly have the same size or real mate pair distance in most cases. The apparent mate pair distance for the large insertions and deletions are different from the typical or average mate pair distance for the bulk of the reads, which we assume are mostly normal in an intact sample. This discrepancy in the apparent mate pair distance can be utilized in the detection of large insertions and deletions. For large insertions, it's possible that one of the mates does not map to the reference genome at all in the case of a completely novel insertion sequence, or is in a completely different part of the genome. This can also be used to detect such structural variations. In a similar manner, one can detect inversions, translocations, and such changes. Here you can see a snapshot of a typical result from large structural variation detection in the workflow. The mix of the deletions and insertions will indicate that it's a complex structure of the sample. An important feature in the workflow is to be able to compare SNPs and InDels between various samples. In cancer biology, this feature allows the user to compare SNPs between paired and normal tissues, between normal carriers and diseased individuals of a family, or amongst families or patients. Based on the comparison, the user will get a flat list of all regions or polymorphisms calls, or SV calls, as the case may be, that are different between the samples that are being compared. In this case, if we compare tumor to blood, from each individual, 4 lists will be generated. 2 of them overlapping region lists and 2 non-overlapping region lists. The regions, SNPs and structural variants that are non-overlapping in the tumor could potentially represent acquired changes that trigger in the tumors. The overlapping lists and those common between the various families will represent inherited mutations which are causes for the cancer. However, the challenge in finding somatic mutation is that these are very rare and requires much more rigorous statistical tests. Now, finding somatic mutations is indeed a challenge. A 2006 study on breast and colorectal cancer says that cells accumulate about 90 mutations in the tumor, but only a fraction of these, about 11, contribute towards actual tumorigenesis. To hunt for these mutations in a milieu of various polymorphisms is not an easy task and requires stringent statistical tests. Firstly, your experimental design requires paired normal samples from several affected individuals. Avadis NGS has a differential SNP analysis feature that runs a statistical test called the Jensen-Shannon divergence test and finds a p-value for the differential expression of the SNPs. You can select the p-value cutoff whether or not you want multiple testing correction, and in case of multiple samples, how many samples need to honor the statistical cutoff. Somatic mutations could be found using this algorithm as those present in tumor but not in the blood. Here is an example that you can see that there is a G to T change in the tumor sample but not in the blood. After you have compared the SNP and the SV lists and identified the lists of relevance to cancer biology, you can further filter these lists and delve into the biology. Let us say you have some prior evidence that there could be large deletions on a few chromosomes, typical in this tumor. Let us say chromosome 1, for instance. You can further filter your reads on chromosome and more precisely the exact location. Similarly, you can filter to see only deletions or insertions that are probably either homozygous or heterozygous in the tumor sample. One can further delve into the data to verify a SNP call. One way to do this is to scroll through the aligned reads in the read track and see the changes. However, in Avadis NGS, we have also developed a variant support view. This allows you to have a compact and insightful view of all reads supporting the polymorphism. In the variant support view, similar reads are clustered together and the number of reads in a cluster are indicated. So if there are sufficient clusters of reads that find the polymorphism, one can conclude that it's real. In this example, we are showing a typical table of results and have selected a SNP that we are interested in that seem to have a fairly good support, 168 in this case. The panel below shows the clustering of reads that support the selected SNPs. The top row shows the reference sequence and the position of the SNP is indicated in the light yellow column. The orange area on either side of the dot with a dot indicates that the read or the cluster is identical to the reference of in that position. And there is a base call if the read contains a base that's not identical to the reference. The first line on the lower panel shows that there are reads that show the variant base in the position of the SNP, but the rest of the reads are identical to the reference. This line, the first line, is indicated as cluster 0. We have effectively clustered 93 reads into one read and represented them as a single line because all the reads are identical in that cluster. The second line indicates that there are 56 reads and amongst these which are all identical except for one position. Here in this particular view, this area is around the SNP is limited to include only 20 bases, 10 on either side. There are various other reads that support the SNP but have differences with each other and the reference as well. The number on the right hand side is the sum total of the number of reads within each cluster. As you go lower down, you can see that there are many clusters with singletons, which likely represent some form of sequencing error and not a short-range haplotype. After you have filtered down a list of SNPs based upon your biological criteria of interest, chromosome, zygosity, and so on, you can investigate all the SNPs which harbor these SNPs. If you want to know, for example, all the genes on chromosome 1 that are affected by homozygous SNPs in the tumor sample, you can click this filtered SNP list and select SNP effect button in the workflow. Typically, if you have multiple unrelated patients, you can now compare gene lists generated from each patient and compare these within experiments. Interesting genes would be those that repeatedly appear to harbor mutations in various patients. Visually, the Genome Browser will let you inspect genes overlapping SNPs of interest. Here you can see a SNP of interest that lies in two overlapping genes, NTRK1 or nerve growth factor receptor, and overlaps with another gene, SH2D2. When we drag the genic effect list or the list of genes that overlap SNPs, we find that the SNP is intronic with respect to all the transcripts of SH2D2 and in the 5' UTR with respect to the transcripts of NTRK1. So a same SNP can have different genic effects on overlapping genes depending upon its position with respect to the different genes. Analyzing sequence data in cancer biology cannot be fully appreciated until one reaches the final steps of the analysis. We can appreciate that there are SNPs in the tumors and they lie in several genes and affect the protein structure. We can appreciate that there are genes with different read counts for their corresponding RNA, and hence these may give rise to different amounts of proteins, some of which may be very important. But how does all the biology of these affected proteins tie up into something meaningful? Avadis NGS provides you with a powerful pathway and go analysis feature to appreciate the real biology in your data. You can build networks and search for known pathways in the lists of genes. To build these networks, briefly, information is extracted from scientific literature which can be used to learn more about the genes that NGS analysis has shown to be affected one way or the other. Strand has developed a technology that can extract information from a vast repository of knowledge that is scientific literature. There are millions of papers that contain tidbits of information that may not have made it to traditional databases like NCBI or EMBL, but at the same time, those are important. These can be extracted using the text mining or the natural language processing algorithm, and they have been put into the pathway database. At the same time, in Avadis NGS, you can load KEGG and other biopax pathways from various databases, search for known pathways, and see which of them overlap your gene list of interest. Here we show you a hypothetical network that's built out of genes from your analysis. The connecting information is developed by text mining. Through the analysis of natural language processing algorithm we have developed in Strand, we can turn a sentence that looks like this. TLR2 expression on monocytes was enhanced by macrophage colony stimulating factor MCSF and interleukin 10, but was reduced by transforming growth factor 1 TGFB1. A graphical representation of the same information would be this. Here the word expression is interpreted as a regulation indicated in the blue box. Enhanced is represented by plus, and the tool also knows about different synonyms for a gene and protein. It can correctly interpret MCSF as the same protein as CSF1. With that, we conclude this webinar session.